Well, thanks everybody for coming to our inaugural first ever Genome, Big Data, and Medicine seminar series. So this seminar series was the brainchild of Pamela Scalar, who's just stepped out, but she'll be back, Judy Cho, who's sitting here in the front, and Orly Bacall, who I believe is not going to be here today. So she's Nature Genetics editor, but has recently taken a sabbatical to be the senior editor-in-chief at Nature Reviews Genetics. I think is off in London for the next few months. But the idea behind the seminar series was one of, obviously lots of talks we're all going to, even within the city, lots of different genomics-y things going on. But we really wanted something that was way more oriented towards how does genomics and multi-omics more generally, the big data that comes from that, how does that ultimately impact human well-being and our understanding of disease, and how is that going to come to play in the hospital of the future when we're all wearing a myriad of devices and being monitored nonstop, but all with an aim towards better maintaining your wellness, better diagnosing and preventing disease. So the idea of this seminar series was to bring in a whole mix of individuals, from those involved in heavy-duty genomics research all the way to those involved in the high-end IT, Google X and Apple. And as you might have seen from the Apple big March 9th event where they released Research Kit and iWatch, we were featured very prominently with the first wave of apps that we generated with Apple to help manage chronic disease conditions. So I think very exciting for us to have a series of these talks from just thought leaders across a spectrum of research in IT and advanced technology. So with that, I wanted to introduce our first speaker, who's Eric Green. Many of you will know as the head of NHGRI for the NIH. So NHGRI, of course, is the largest singly focused on genomics research organization in the country. So a lot of you here likely benefiting from grants coming from NHGRI, but of course they have a strong intramural program as well. And Dr. Green, of course, was part of that before he became the head of that institute, having driven, was chief of the technology arm, was a key component to driving the sequencing of the human genome, of understanding eukaryotic genomes in general, so maintained a very healthy research program over the last couple of decades, in addition to leading a lot of the different technology and sequencing arms for NHGRI before ultimately taking on the top spot. And of course, you might have all seen the big precision medicine talk that Francis Collins gave, and that talked about Obama's vision for instituting precision medicine throughout the country and funding that kind of stuff. And that's something that, of course, Dr. Green played a key hand in. And so today he's going to talk to us about the genomics, precision medicine, and how this is going to impact our understanding of health. So please join me in welcoming Dr. Green.
Well, thank you for that kind introduction. I'm delighted to be here. I hadn't appreciated until a few minutes ago that I'm the first lecturer in this new series. And in many ways, I think that's terrific, because actually what my talk is going to do, I think in many ways, is going to set the table for subsequent talks by really describing in a very rapid form sort of a landscape view of where we are in human genomics. When I originally got the invitation for this, I was delighted to participate, and I had originally envisioned, because this was a couple months ago at that point, I had originally envisioned spending the entire hour sort of setting this view of what has happened and where the field of human genomics is going, especially with clinical applications. But there has been a significant new development with this Precision Medicine Initiative announcement, and I really want to share that story with you. So actually, I've truncated the first part of my talk, it'll be about 60% of my talk, really describing this landscape view of human genomics. And then I'm going to spend the last 40% or so of my talk telling you the story of this Precision Medicine Initiative up to everything I know to the present time. And it's very rapid and developing, and I wanted to share it with you. So I think especially for the students in the audience and trainees in the audience, I really should remind you that the field of genomics is an incredibly young one. It's really only about a quarter century or so old, which for a biomedical discipline is actually quite young. In fact, the first use of the word genomics in the scientific literature came in the inaugural issue of a new journal called Genomics, actually, and in this lead editorial, it was described what this new field was. It's a significant year for me. It's the year I graduated with an MD PhD degree, which meant that throughout medical school and graduate school, I never heard the word genomics once, and that was because the word didn't really become common use until 1987. But the reason this became a significant word in 1987 is it was in the later phases of the intense planning that was going on to launch what arguably is one of the most significant projects ever undertaken in biomedical research, the Human Genome Project, which I had the fortune of being able to participate in on day one, starting October 1 of 1990. And in fact, we're coming up on our 25th year since the beginning of the Human Genome Project. But in many ways, while this project was unprecedented in its scope and its scale and its organization and its international flavor, it was certainly worth celebrating what it had accomplished. In many ways, it was just the starting line for everything that's unfolded in the 12 or so years since. And in fact, I think all of you are well aware that genomics is just everywhere these days. In fact, the applications of genomics are wide and far, and in fact, all the areas shown here, each one of which could be a talk in and of itself, and I'm really not going to talk about any of these areas except to simply note how important genomics has been to advance these areas. What I'm going to emphasize in my talk really are the applications of genomics to health and disease and medicine. And of course, the reason that's going to be my emphasis is the place I've now worked for 20 years, and that is the National Institutes of Health, with clearly a very laser focus on turning discovery into health. But in particular, I've been at the Institute, as Eric mentioned, at the National Human Genome Research Institute, which, keep in mind, was created by the U.S. Congress to lead the U.S.'s effort in the Human Genome Project, but of course, since then has expanded its mission to really now take on the role of trying to advance human health through genomics research. I started at the Institute 20 years ago as a tenure-track investigator and have had various positions along the way, and then just a little over 5 years ago became the director of the Institute. And I'd say more than anything, I feel my role in leading the Institute is to help navigate it into a position where, in addition to studying how the human genome is put together and how it works, is thinking about how to apply this so that we can improve the care of patients using genomic information. And so the framework I tend to think about is a framework which I call genomic medicine, which definition is given there, and I think should be familiar to many of you, just recognizing that each of us and each of our patients has a unique genomic makeup, and if we can get genomic information about patients, we clearly should be able to improve their clinical care. Again, largely synonymous with personalized medicine, individualized medicine, and as you'll hear later, precision medicine. I keep the focus, especially for our institute, on the genomic aspect of individualizing care, so I use the phrase genomic medicine. Now, in thinking about how are we going to realize genomic medicine broadly defined, I would sort of liken it to a long journey. And it's not going to be a simple journey. The starting line was the Human Genome Project, and the finish line, if you will be when we have large-scale deployment of genomic medicine. But we have to recognize this is going to involve many, many steps, many of which we know, but many of which we probably don't know. And furthermore, it's not going to be a journey that's going to involve one discipline or another discipline or a small community of scientists. Rather, the analogy I use is going to be much more like a marathon, where many, many people, many different disciplines are going to have to be brought to bear, running shoulder to shoulder for a very long time to really figure out how we can use genomic information to improve human health. So how do you take on a challenge like starting with the human sequence that was delivered by the Human Genome Project and actually change the practice of medicine? Well, that's a Herculean task, but you have to start somewhere. And the way we tend to always start in genomics in thinking about a challenge like going from base pairs to bedside, or if you prefer the metaphor, from helix to health, is think about strategically what needs to be done for the next few years, describe that in detail, and use that as a guide for what needs to be accomplished. And this is exactly what we did during the Human Genome Project. We actually did it multiple times. We'd come up with a plan. We'd execute the plan for a few years. We'd look at the plan again. We'd say, it's a little old. We'd rip it up. We'd come up with a new plan. So it wasn't surprising that the day the Genome Project ended, our institute published view of what needed to be done, a vision, if you will, for the future of genomics research, now having completed the Human Genome Project. And that worked pretty well for a number of years, and then of course it got a little gray because of some substantial advances in genomic capabilities. And so now, actually, almost 4 years ago, we published what is our current strategic vision for genomics. And it was the one that was published shortly after I became director. And the signature thing about the current strategic plan that we're operating on is the overt reference to genomic medicine, the overt indication that it was very clear that now was the time to start thinking about how to apply genomics as part of medical care. So what I want to do in this first part of the talk is really describe to you in highlight form really what has transpired that is now, I think, beginning to bring genomic medicine into focus. And there really are probably about 5 major areas of our accomplishment that I really wanted to highlight because I think and I'm sure other speakers will dig deeper into some of these areas because it really has been some remarkable advances in these areas. The first area, of course, relates to simply understanding what the human genome sequence actually does, how it codes for information. And of course, that had to become a very key part of what genomics was doing as a scientific discipline because this is actually what the Genome Project delivered. Of course, it's only 0.0001%, but you get the idea. It just delivered the sequence. It did not deliver an interpretation of the sequence. And so that's why literally, actually even before the Genome Project ended, tremendous efforts went in to figuring out how it was that these string of letters actually encode biological information. One of the ways to jumpstart that, of course, involves sequencing the genomes of other related species and using the power of comparative analysis to illuminate those parts of the genome that are under high or heavy evolutionary conservation, signaling regions of the genome that are likely functionally important. We did lots and lots of other mammalian genomes and quickly found out that something on the order of 5 to 10% of the human sequence is conserved across almost all mammals, which gave us a place to really look more carefully as to what the functional significance of those sequences are. We learned that among those sequences, as we tuned better and better computational methods to figure out exactly where were the genes and where were the other elements, we can quickly started to be able to catalog what were the sequences directly coding for protein coding genes. And we learned after some refinements that there's something like 20,000 genes in the human genome, but that those genes only reflect about 1.5% of the letters of the human genome, leaving a significant fraction of the most highly conserved sequences doing something other than directly coding for proteins. We now know that those remaining sequences are incredibly important biologically and quite complex in addition. They, of course, are all the regulatory sequences and sequences involved in chromosomal dynamics like chromosome replication, segregation, and so forth. But we trail way behind understanding the full complement of these non-coding sequences. Oh, and I think the other thing that clearly has come to the fore over the last 12 years in particular is the recognition that going beyond the primary sequence of the human genome, there's all this epigenomic language that's going on where parts of the DNA need to now be highlighted, signifying regions of DNA that are getting associated with different proteins to signal information and also getting decorated with various modification groups that also clearly are biologically important. And so the epigenomics landscape is also something that has to be elucidated. Recognizing the need to catalog all the coding functional regions, non-coding functional regions, epigenomic marks in the human genome, that is the reason why NHGRI launched a major program called ENCODE for Encyclopedia of DNA Elements, which basically has basically since almost the end of the Human Genome Project, has in consortium form been trying to develop computational data sets and biologically derived experimental data sets that basically provide richer and richer and richer annotations of the human genome. Shown here is one particular region of the human genome, and you're overwhelmed with all the horizontal tracks depicting everything from where the conserved sequence is to where regions of open chromatin, where the epigenomic marks, where a gene, so on and so forth, giving you, if you will, an increasingly better and better GPS-like view of the genome in terms of being decorated with various annotated information base by base across the human genome. And I would also call your attention that ENCODE is not the only effort that is trying to elucidate all this functional information about the sequence. And you may have seen this recent issue of Nature where the epigenome roadmap effort, which was a major multi-institute common fund program at the NIH, delivered a series of papers, again, describing a lot of information about epigenomics and the human genome. So, where are we 12 years after the Genome Project in understanding the real interpretation of the human genome? Well, we're probably about here. I mean, we used to be here, but got to be here. And I'm sure for decades from now, we'll still be interpreting, reinterpreting. We have an incredibly good catalog of at least the genes and increasingly non-coding elements, but we're a long way. We will be doing this for a long time. And I think our thirst for knowledge about how the human genome works will absolutely be driving this forward. One of the things, of course, that has helped these studies and many other studies I'm going to talk about that I think also deserves to be among the top 5 things that have happened since the end of the Genome Project, of course, are the development of new genome sequencing methodologies. Don't take these for granted. For those of you who are new to the field of genomics, this was not guaranteed the day the Genome Project ended. But nonetheless, the day the Genome Project ended, in the strategic plan I alluded to earlier, was the cry that we needed better technologies, perhaps ones that would deliver the sequence of the human genome for something like $1,000. And the reason why that was so audacious is the day the Genome Project ended, we had sequenced the first human genome, and that came in at a cost of something like $1 billion or some number of hundreds of millions of dollars, depending on how exactly did cost accounting. But we recognized this was not a practical way to sequence human genomes going forward, certainly not for clinical applications of genomics. And so we needed technologies to be developed that would lop at least 6 zeros off of that figure and eventually delivering what became the battle cry for the community of the $1,000 genome. Well, in many ways, the rest is history. We had a major granting program. Private sector got involved in this as well. And as has been written about in Nature or chronicled on our website in this sort of iconic-like graphic, the costs for sequencing human genomes have plummeted. With the advent of these various next-generation sequencing platforms, one after the other, each with their own nuances, but overall have seen a plummeting in the costs of DNA sequencing, such that now we can affordably sequence human genomes, even though we want the cost to come down further. Keep in mind, it's not just the cost, it's also the amount of time. Just as a reminder, that first human genome sequence, which I told you about, was something like $1 billion. There were 6 to 8 years of thousands of researchers around the world working to actually generate that first sequence. And of course, that's a long time to get a human genome sequence. But today, using any various methodologies, you can get that sequence in something like a few days with expectations it'll be down to a day shortly. The cost, it depends exactly how you do your cost accounting, is not quite $1,000, but is certainly on a trajectory to get us to $1,000 or below, perhaps even in the next year. And of course, whatever technologies you think you're using today, it's likely going to be different technologies tomorrow, and lots of eyes are on these new devices that use nanopores, including a device like this, which plugs into the USB port of your laptop computer, and allegedly, hopefully with some refinements, will deliver a human genome sequence in something like a day for hopefully less than $1,000, and we're very excited to see how this technology progresses. I was particularly impressed when I asked the manufacturer of this particular device about the laptop computers. The person guaranteed me it would work equally well in a Macintosh or a PC, and I thought that was reassuring, and I'm going to hold him to it because I think it's important. Now, but when you can start to sequence human genomes for $2,000, $3,000, $4,000 or $1,000 or less, what quickly becomes the next big hurdle is actually dealing with all the data. And I think all of you recognize that the new bottleneck we have, which we absolutely did not have 12 years ago, is basically being victims of our own success. We have these technologies that spew out data so incredibly prodigiously that it overwhelms us. It is like drinking water out of a fire hydrant, out of a fire hose. And now, all of a sudden, we've gone from a situation where all we wanted was data to being overwhelmed by the kind of data that we have. Now, this is not unique to genomics, but I think genomics is the poster child for this. We have sort of arrived in the big data era for biomedical research, and genomics has certainly driven us there along with other technological advances in other fields and other disciplines. In the case of genomics, though, it really is something that is a bottleneck. These fancy methodologies truly can read out a human genome sequence in something like a day or 2 or 3. And we can sort of slog away, but we need, and we can, absolutely get to that list of variants that exists in any given patient. But it doesn't come without pretty big hurdles. We have hurdles related to hardware, just having to be able to store all the data, push the data from site to site through the internet, and just processors to be able to just harness all the analytical tools that are needed to analyze the data. All sorts of issues about software and software availability, including to casual users, not just the experts at the best genomic centers in the country. And of course, we need better software tools. Trainees in the audience should take note that there's a big workforce issue. I can tell you that those of us at NIH are very worried about, are we training people correctly for this big data era that biomedical research is going to face? And of course, specifically around the variants, do we really have the tools in place to really understand what a given variant means in a given patient? And of course, the answer is not yet, although we are working on that. Recognizing this big data era that we're in, genomics driving it, but other areas driving it as well. I hope all of you are aware of something I was involved in trying to orchestrate at the corporate NIH level, was this new initiative called Big Data to Knowledge Initiative, which has many different components. If you don't know about this, it is now completely launched. Just a couple years ago, it was just an early idea, but this is an example, even in fiscally tight times, of NIH recognizing that we had to do something to sort of catalyze growth to deal with this big data situation we're in. And even with tight resources, we figured out a way to make this happen at the corporate level through various means. And I would point you to read a little more about the Big Data to Knowledge program if you don't know about it. So we have better understanding of how the human genome works. We have better technologies for sequencing genomes and analyzing genomes. That really gave us the ability to now start doing the kinds of things we needed to do to really apply genomics to understand disease and eventually improve medical care. The third area, of course, then involved trying to have a better cataloging of how all of us are a little bit different, how our patients are a little bit different in terms of their genome. And of course, while it was known for a long time that any two individuals differ by about 1 out of 1,000 bases, so we can start decorating any given human genome with these for variants, at least at an individual base level. Of course, there's other kinds of variants as well. It was recognized that the great majority of those variants are inconsequential from an individual point of view in terms of their health and well-being. But a subset are consequential, some of them being bad variants in terms of conferring risk or overt cause of disease, and some being good variants, maybe being a protective variant. But it turned out that it's not that everybody has their own unique set of variants. It turns out that most of the variants you have, somebody else in this room already has as well. And it would be great if we could catalog the more common variants that are out in the worldwide population to set up a circumstance where geneticists could study them and figure out which ones should we not care about, which ones are risk conferring, and which ones are protective. That's where we want to be, and that seemingly became something that could be tackled with a large-scale program. So there were programs like the SNP consortium that tried to catalog single nucleotide polymorphisms that gave rise to the HapMap project, which tried to get more data about variation, including the relationship of variants relative to one another on chromosomes. And when the new technologies for sequencing came along, it was very clear it was time to launch something even more audacious called the 1,000 Genomes Program, which attempted to collect individuals, originally 1,000. Once again, geneticists are overachievers. We had collected over 2,500 individuals from a discrete worldwide populations, 26 in total, and then just start sequencing them and make available all that data on the internet so we can start to catalog the more common variants that exist across the world. A pilot project was published in 2010. The next phase was published in 2012. And coming to a major journal near you later this year will be the final installment of the 1,000 genomes in terms of their large-scale variation data. We went from a circumstance 12 years ago, at the end of the Genome Project, of maybe having tens of thousands of variants that we knew existed, that we had available for research to study, to now having close to 100 million. This is the kind of thing that now sets up a circumstance where people can start to study which of those 100 million variants are consequential, which are not. That becomes substrates for a whole host of studies that I'll be discussing a little bit in a minute. But the other thing 1,000 Genomes started to do was to also start to give us an idea of the kinds of things that we can know about in terms of an individual patient. When we actually sequence an individual's genome, what does it look like? So if you're not familiar with these numbers, it's fun to go through them. Each of us and each of our patients has about 6 billion nucleotides. You got 3 billion from mom, you got 3 billion from dad. We're all diploids. If it turns out when you actually sequence people's genomes, you do in fact find about 3 to 5 million single nucleotide variants compared to any given reference in each individual person. When you start to analyze those variants, you can start to learn, oh, by the way, those variants, as we expected, are mostly in common with other or already known in databases. In fact, any given person you sequence, you only find about 150,000 of those variants have you not seen before. All the rest of the ones are already in databases. And actually, it also turns out that each of us has about 60 variants that neither of our parents have. So this represents new mutations that happened in the process of creating you. In terms of what's known about the biological consequences of these variants, well, here, of course, we know most about how it affects protein coding regions. And it is now clear that each of us carries about 1 to 200 disruptive variants that kills one of our genes, at least one copy of one of our genes. And interestingly, on average, each of us has about 20 genes where we're knocked out for both copies of our genes. So out of our 20,000 genes, you're basically a homozygous knockout for about 20 of them. And some of that is consequential, and some of it is probably not consequential. But at least it gives you some idea of the range that we now can expect in terms of when we start to sequence patients' genomes. Of course, that only incentivizes us to try to now understand what exactly is the relationship between all those variants that are sitting in each of us and actually what role they play in disease. And so elucidating the genomic basis of disease became a very major opportunity with better and better technologies, more knowledge about variation, and so forth. Now here, of course, there's sort of two parallel stories, although in many ways they start to converge. But the easiest way to think about this is just to remember the simple dichotomy between rare diseases and common diseases, although sophisticated people in the audience knows that there's some blurring between that. But by and large, on the one hand, we have rare diseases that are genetically simple, like cystic fibrosis, sickle cell disease, and so forth, where it's basically a defect in what is typically a single gene that is the major risk for getting the disease. There might be some environmental contribution. There might be other variants that in some way influence morbidity and mortality. But by and large, it's a single gene Mendelian disorder as it's known. Well, this has been greatly accelerated in terms of finding the basis for these single gene disorders with better methods for sequencing, better annotations of the human genome, more knowledge about variants. And in fact, just to give you two numbers to keep in mind, the day the Genome Project started, there were 60 rare diseases for which we knew what the mutated gene was. We knew the molecular or genomic basis for 60 rare diseases. Fast forward to today, and that number is nearly 3,600. So a substantial increase. Of course, there still remain about another 3,700 rare diseases that we still need to find the defective gene, and those efforts are going on through a number of programs, including some programs that we fund. But substantial progress has clearly been made. Now, that's in contrast in terms of how this is approached to common diseases, which, of course, are the diseases that fill hospitals and clinics to a much greater extent than rare diseases. But of course, common diseases like hypertension and diabetes and cardiovascular disease, mental illness, and so forth also have the challenge of being genetically complicated because it's multiple variants that are influencing the risk for disease, and in most cases is a greater contribution of the environment. This, of course, has been greatly studied and renewed in many ways in terms of momentum with all the available information about common variants through efforts like the 1,000 Genomes program. That has led to the genome-wide association study era, the GWAS era, which really the first success story was published in 2008, and now there's over 2,000 publications describing successful genome-wide association studies. These deliver information about where to look more carefully in the genome, but don't deliver necessarily the causative variant, and in most cases, almost in few cases, to do that just delimits the space. What's been very clear from these studies is that we know a lot about hundreds of common diseases in terms of where in the genome likely harbor these variants, but teasing out the exact variant and figuring out how it confers risk is turning out to be very challenging, probably will require larger studies, inevitably is going to require genome sequencing on a larger scale and done in a way that is going to have to generate a lot more data than has been available by the current GWAS studies for most diseases. But nonetheless, I think we really have moved the ball down the field considerably in the case of common disease as well. But of course, this all just gives us information sort of about discovering variants and how they play a role in disease. I've said nothing so far about any clinical applications. And that is, in fact, my fifth area that I wanted to highlight. And of course, that's actually beginning to implement genomic medicine. And by that, I mean truly doing things that changes the course of clinical care of patients. And here, what I would say is I probably don't have as much progress to report compared to some of these earlier areas. But what I would tell you is I think it's very clear that there's some hot areas that are emerging. And in fact, these are the very areas where I can tell you my institute is funding to try to accelerate progress. So I thought it would be fun just to quickly tell you about some of these programs, but also to highlight what I think are sort of like the 6 hottest areas, if you will, in genomic medicine. And in each case, we have a footprint in terms of funding programs. Certainly, I think the hottest area in genomic medicine is one I really haven't talked at all about, and that's cancer. Cancer is a disease of the genome, and it's very clear that the applications of genomics to improve how we take care of cancer patients has come to the fore for some specific cancer types in a very convincing fashion. It is now sort of a circumstance where I think we will begin to see the introduction of genomics as a standard tool for cancer care sort of becoming just sort of standard. I mean, just once upon a time, we introduced radiographic imaging as part of standard clinical care. I think increasingly the kinds of genomic maps we make from analyzing the genomes of tumors that show all the derangements and having that serve as a fingerprint of how to take care of that patient will become mainstream for some kinds of cancer, and I think you'll see that number will increase in the coming years. And in fact, all you have to do is look to see how cancer treatment is advertised in a competitive market. There's lots of examples of this. This is just one organization, and you can see if you go to their website, I mean, it's just genomic everywhere, genomic this, genomic that. This is now regarded as sort of commonplace for offering genomic approaches to cancer care. I think the other low-hanging fruit, although there's some early examples, many more are needed, of course, relates to pharmacogenomics, where we clearly have learned more and more about how genomic variants play a role in drug response, and perhaps we can better get the right drug to the right patient at the right dose by using that information to stratify patients before we ever decide what their medication is going to be. And there, again, some successful examples, and certainly FDA has more and more medications where they request the labeling to be put on that drug saying genetic genomic information might be helpful in the use of this medication. And we think that list is going to grow with time. We have a number of projects and programs to try to accelerate progress in pharmacogenomics. A third, I think, hot area certainly relates to very rare genetic diseases, in particular genetic diseases that have been undiagnosed. This has been chronicled in a number of ways, such as here in Nature last year, writing an article where you have children born, have no idea what's wrong with them, and indeed it just starts talking more and more. It makes sense to sequence their genomes as you're doing this extensive workup that is not necessarily revealing clues about what's wrong with the child. This was in particular sort of exemplified by a program that was originally started, the intramural program at NIH called the Undiagnosed Diseases Program. Here's Bill Gall in the center, who is sort of the founder of that program. And that program was so successful to take patients from actually around the country, where it had gone from medical center to medical center, no diagnosis, bring them to NIH, work them up, including genomic sequencing. And in something like a quarter to a third of the cases, you were able to figure out what was wrong with the patient from that kind of a workup. Based on that, this program has now been expanded. It's created a national network called the Undiagnosed Diseases Network. The initial set of sites are shown here, and they're just in about their first year or so of organizing as a network to basically have a number of national facilities doing this kind of approach for undiagnosed cases. Now, inspired by some of the early wins in cancer genomics and pharmacogenomics and rare disease diagnostics, I think it became clear to us that we needed to sort of expand our scope by really figuring out how genomic medicine could be used on a larger scale for clinical medicine. And so we started what I call test drive programs, where we really gave out grants to really use genome sequencing and genomics as part of clinical care. You should be familiar with two consortium that we now have going, one that's in about its fourth year, called the Clinical Sequence and Exploratory Research Network. The sites are shown here. And in each of these cases, some related to cancer, some related to pediatric disease, some related to pharmacogenomics, real cases of clinical care being influenced by genomic information of patients so that we can get some early lessons and develop some early best practices. Recognizing that we want, of course, the successes not to just be limited to major academic centers, but also want to make sure that information is disseminated more broadly. We also recently started a consortium shown here called the Ignite Consortium, where in this case, including one of the grantees at this institution, is attempting to sort of to establish ways to disseminate best practices in genomic medicine to different healthcare delivery systems so that everybody can benefit from genomic advances. In addition to our test drive programs, other programs that I think are really representing hot areas in genomics relate to both prenatal and newborn periods in terms of ones where genomics is clearly being applied. In the case of prenatal, non-invasive prenatal genomic analyses is really becoming one of the hottest areas, I think, in genomics right now, in particular because these methodologies clearly have been developed and are being deployed commercially on a very large scale, being recognized by the popular press as having some really important technologies and leading to lots of questions and also some challenges, scientific, also some ethical questions about, but nonetheless, this is really gaining a tremendous amount of momentum and represents sort of the ability of using the power of these methods for sequencing DNA to detect fetal blood floating around a maternal bloodstream at very high sensitivity in a way that means you can just do a simple blood draw instead of more invasive procedure to do prenatal testing. At the other end of gestation, of course, you have newborns, and here lots of discussion, including by Time magazine, about what the future is going to bring in terms of genome sequencing and genomic analyses of newborns. Recognizing that this was going to be an important area to learn more about and wondering not only logistically, would we ever consider a circumstance where we sequence the genomes of newborns, and if so, what are some of the ethical, legal, and social issues associated with that? We launched a program jointly with the Child Health Institute at NIH called NSIGHT, and you can see the long name there. The first 4 sites are shown here, and each of these sites is looking at the problem from a slightly different perspective, but is sort of getting us the kind of research we need to make informed decisions about what this is going to look like in the future. In particular, the site at Kansas City, I think, has really demonstrated, as talked about in Nature this year, that in the case of very sick patients in the NICU, where basically you have no idea what's wrong with this child, would likely expire in a matter of days, fast sequencing of their genomes has in some cases revealed some important clinical information that can actually aid their care. And so that center in particular is looking at sort of what all the parameters around the idea of doing genome sequencing in the case of a neonatal intensive care unit. And finally, I should tell you the last area is one where I think we need more progress, but I think is a very hot area in terms of importance. Of course, that relates to just understanding what all this genomic data really looks like. You know, we sort of have a vision that, wow, we can get to the point, and we are technically, we can sequence any patient's genome, but it's not going to be a successful story if we round on that patient in the morning and this is what we look at, and just wonder what the sequence actually means. And so recognizing the importance of this area and also recognizing that we need systems that can aid healthcare professionals to interpret the information coming out of genomic analyses, many of which are going to be integrated very nicely with electronic health records, and also are going to need to be accessed through very convenient devices, tablets, iPhones, and so forth. We need to develop in the knowledge base to be able to inform healthcare professionals what these variants mean and what you do about them clinically. And that is why we created a consortium called ClinGen, which aims to develop that knowledge base, if you will, to try to help practicing healthcare professionals understand all the variants that are being reported in the literature, but what you do about them clinically. And we need all new analytical methods to figure that out, considering the tsunami of advances that are coming out every week with respect to correlating specific variants to possible clinical actionability. So that's the very rapid form sort of painting of this landscape that I wanted to share with you. I do want to just point out a couple of other things that I think are really relevant to what I'm going to talk about in the second half of my talk. And that is that I've sort of just quickly romped through about 25 years of genomic progress. And when it started and I got involved in this field, it was really about just researchers at the bench, researchers at computer terminals. It was a biomedical research endeavor. And, when the Genome Project ended and we started to see clinical applications, increasingly I think healthcare professionals did get interested in genomics. But still, it was a professional endeavor. I think the difference now with cancer in particular and some of these other applications that were hot that I told you about is that genomics is becoming mainstream for patients, and certainly will by 2020. And if it's mainstream for patients, it's going to means friends and relatives of patients, which now means all of us. And so I am completely convinced that one of the other things we all need to do as a community is to start to recognize that the relevance of genomics is basically societal relevance. And we also have an obligation to help foster the understanding of genomics in the general public so that when people go get their medical care over the next 5 years and beyond, they understand what the physicians are saying to them and the other healthcare professionals are saying to them. Now, that's my theory. Is it right? Well, I think it is, and I'll just give you a couple examples. If you go to the newsstand, these stories like Angelina Jolie's story has brought genomics very, very much in the consciousness of the general public. And even things like Time magazine had a special issue over the summer talking all about DNA and how it shapes our lives and so forth. So the newsstand sees genomics very prominently featured. Similarly, our family room sees this. If you put your television on or you put your radio on, I don't know about the New York market, but I can tell you the Washington, D.C. market, we have advertisements all the time on TV and radio, such as this one.
Some say your health is the luck of the draw. But Innova's groundbreaking genomic research is working to change that. We're studying the genetic makeup of infants to detect if, when, and how disease may occur, so we can learn to tailor the best care for each patient. Providing individualized health care, anticipating and preventing illness. That's predictive medicine. Innova. Join the future of health.
Now, I'm not endorsing the commercial. In fact, there's some aspects of the commercial that can be paused. But whether you like it or you don't like it, it doesn't matter. This is what I think the general public is going to be increasingly confronted with, whether it's cancer treatment centers or whether it's hospital networks, that genomics is going to be talked about. And I think we also have to be thinking, therefore, about our responsibilities as a society in educating, explaining, and helping to disseminate this information so that the communication channels are clear and are robust and are effective. And so that's what I want to tell you, sort of in painting this very broad landscape about genomics. And I can't help, especially with some of these last things I was talking about, sort of point out that it was very clear that there's a lot of growing interest and a growing relevance of genomics in the general public, and also in thinking about sort of what needs to be done to sort of keep the U.S. at the cutting edge of scientific advances. So let me transition now and tell you about a recent story that has been very fast-paced. It has consumed a lot of my life, actually, for the past 9 months, beyond my day job. And I just want to tell you, and it's about this thing called the Precision Medicine Initiative. There is a story behind it that I thought would be fun to share with you. And then I'll tell you what the vision is, and then I'll tell you about where we are right now in terms of planning. So let me start with the story. And one thing I'll tell you about the story is that it's been really hectic. And the reason it's been really hectic is because it has a lot of very influential people, in particular one who's been sort of spearheading this. And the individual who's spearheading this really is the president. And I don't say that lightly. I am literally meaning, and I'll prove to you, it really is the president who's driving this. Now, President Obama has been interested in research for a very long time. In particular, though, he really likes genomics. When he was senator, then Senator Obama in 2006 actually introduced a bill in the U.S. Senate called the Genomics and Personalized Medicine Act of 2006. Turns out that this bill didn't get much traction, didn't get passed, but nonetheless it signaled his interest in personalized medicine and genomics. Now, it also should come as no surprise that when he became president, he did surround himself by people who also are very interested in genomics. He appointed Francis Collins as head of the NIH. He appointed Eric Lander as co-chair of the Presidential Council of Advisors in Science and Technology. And both of them regularly get to interact with the president and tell him about science. He also has some major friends in the private sector and the genomics industry that also, I think, has influenced his thinking and excitement about genomics. And he basically also has become aware of some exciting genomic opportunities in terms of medical applications. And so actually in June of last year, 2014, he actually called a meeting in the Oval Office that included people like Francis Collins and Eric Lander to talk about opportunities in genomics and personalized medicine and so forth. And he got so excited about what he heard that he basically said, I want to launch a program in the last 2 years of my presidency in this area. Bring me a plan in the fall. And now when he discussed it, and as it got discussed a little further with people in the White House and beyond, it actually was appropriately cast in a slightly wider context, recognizing that it wasn't all genomics, but there also were issues like environment and lifestyle and so forth that were also important, and the framing of precision medicine was the phraseology that got used, which in many ways is just a broader context for individualizing medical care to advance human health. I told you about genomics early on and genomic information, and that's one piece of the puzzle. Precision medicine is just broadening this to include other things like life cycle, environmental exposure, diet, and so forth. Now, much of this, I admit, was influenced probably by this National Research Council report from several years ago on precision medicine, which in many ways put the phrase very much at the front of many people's thinking. Now, it really actually is a relatively simple concept, but the president really liked it. The idea, of course, is that, today, it's true. I'm a physician too. Most medical care that we deliver is based on the response to the average patient. But we recognize, and genomics is certainly one of the main areas, that, we want to live in a tomorrow where we really are basing care on differences in our genomics, our environment, our lifestyle, and that's going to simply provide a more precise way to take care of patients. But how do we get from this today that we're living into a tomorrow? Now, meanwhile, it's not like this whole idea is new. It's not like physicians haven't attempted to be precise. Of course, we try to be precise. In some cases, we use things like prescription eyeglasses for many centuries. That's a personalized way of approaching a more precise way. We've done precise blood transfusions for over 100 years because we had the tools available to do so. But of course, what we need is something bigger and bolder to try to bring many more things together to come up with many, many more examples of where precision medicine can be used. And what the president basically wanted was something to be very rigorous in terms of research program to accelerate this. Recognized it needed to be multidisciplinary and was not scared about it being audacious in scale and scope. And he basically said, bring me a plan that would allow us to jumpstart precision medicine at a national level. So that basically created a circumstance in June where NIH was asked to lead. Other federal agencies are involved, but NIH was taking the lead pretty much in trying to develop the plan and at least coordinate the planning for this and to try to deliver something by fall. And it consumed a small number of us at NIH our summer throughout July and August and September. By October, the president was reconvened the group in the Oval Office. This is actually a photograph from that meeting. You can see Francis Collins is there, and Eric Lander. John Holdren is his science advisor, and major other members of the White House and his cabinet listening in. And he loved the plan. He thought this was incredible. He definitely wanted to pursue something in this. He said, okay, build this into the fiscal 2016 budget. The president announces the budget in early February. Usually talks about things in late January, including the State of the Union address. And he said, build it into the budget so I can announce it in early 2015. So by November, we got word that indeed not only was the president going to put this in the budget, but that he was actually going to announce it in the State of the Union address in some way. And that created great excitement, and we even celebrated briefly and then went back to work. Because it meant that we had to get ready for a rollout of this and have all of our ducks lined up and getting ready for whatever this was going to look like. And so that took us up until December. January rolls around, and then the State of the Union address. I will tell you that we did not know when the State of the Union address was going to happen exactly what the president would say. We had heard of different language, sort of a reference maybe to precision medicine or maybe a more overt announcement. And so we waited along with everybody else to see exactly what he would say. It was a well-kept secret. And then January 20th came.
21st century businesses will rely on American science and technology, research and development. I want the country that eliminated polio and mapped the human genome to lead a new era of medicine, one that delivers the right treatment at the right time. In some patients with cystic fibrosis, this approach has reversed a disease once thought unstoppable. So tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. We can do this.
So that was way cool. He actually announced it. It's particularly cool because I know you politically savvy folks recognize that there are very few times that you would see Boehner and Biden both clapping at the same time, let alone people on both aisles standing up at the same time. So this was good. So there seems to be bipartisan support, which is going to be needed. I want to share one other video with you because, again, you wouldn't know about this because unless you just happen to be there or happen to look at this video. We had been told, those of us who basically gave up half of our summers working on this, we had been told that really the president was into this. He was really into this. He loved it. But this is coming from Francis Collins, who's my boss, and, he's sometimes a little manipulative, makes me work hard. Just, okay. Sometimes over the top. But it was an interest, but I believed him. I've worked for him for 20 years. I believed him. But the fact is, it was very interesting. So this is January 20th, the State of the Union address. It just so happened that January 28th, the Secretary of the Department of Health and Human Services, so this is Francis Collins' boss, and so this is where NIH lives, the Department of Health and Human Services, Secretary Sylvia Burwell, who's new to her job, in less than a year, came to NIH. A couple times she tried to come and had to keep canceling. This was finally her visit. And she actually came and gave a town meeting in our biggest auditorium on the NIH campus for the NIH staff. And she took questions that were emailed in. And one of the questions, this was unscripted, one of the questions that came to her was, tell us a thing or two about the President of the United States, because you worked with him very closely in the Office of Management and Budget, which he previously had, and now as Secretary of Department of Health and Human Services. Tell us a couple things about him that the NIH staff would like to hear. We had no idea what was going to come out of her mouth, and this is what she said.
But with regard to a couple things about the president, two things that I think are important to know that could help you in your day-to-day work. The first one is, and I'm sure you've told the team this, the precision medicine thing, that's him. This is like a presidential priority. This is the amount of time that is being spent on this issue by the President of the United States relative to, I could go through a whole list of issues, is incredible. And so what you should know and what you should take from that is this is his own personal interest. It is what you all do. It is, he believes so much that this is about our nation's innovation in science. He believes this is about our economy. He believes this is about the health and welfare of our people. And so this, while it's precision medicine, it is I think it gives you a view into his thinking about the work you all do every day.
So that was January 28th, and all this was organized in a fashion so that before the announcement of the budget, which was going to happen the next week, on January 30th, in the East Room of the White House, came this announcement where he now formally went through in very great detail what the initiative was going to be about and what the justification behind it. And so this was the formal rollout, if you will, announcing the initiative. We stood back, curious what this was going to bring in terms of the press. Generally, very positive press coverage in terms of the popular press. And similarly, I'm sure many of you saw not only this, you probably also saw the scientific press covering this as well. Again, generally quite positive, at least what they knew about it. So that was the story. Let me briefly tell you about the vision. This was all sort of the fun story leading up to the announcement. Coordinated with the president announcing on January 30th the initiative, that very hour that he made that announcement, it had been arranged so that published online on the New England Journal of Medicine's website was this publication written by Francis Collins and NCI Director Harold Varmus that described in a little more detail what exactly this initiative was going to be about. And if you have not read this, I would strongly urge you to take a look at this. Let me just briefly introduce you to what the initiative is envisioned to be. It's envisioned to first have a priority area of sort of a near-term win, if you will, by using cancer as a model of precision medicine, not surprising based on what I told you earlier. Recognizing that cancer is at the leading edge, but we could ramp this up and simply be more aggressive in terms of being able to tackle additional applications of genomics to improve cancer care. And so that's going to sort of be regarded as a near term. But also recognizing that we want to move beyond cancer to expand that model to other diseases, was recognized that what was needed for that was the creation of a national cohort of over 1 million research volunteers, using that to generate a knowledge base for precision medicine. Other things were going to be needed, including policy changes and using the president's announcement as an opportunity to try to remove some barriers to clinical implementation, both regarding issues related to protecting research participants and also issues related to FDA's oversight of precision medicine products. So that's the vision. I'm going to tell you a little bit more in a second about it. I should stress that in the president's budget, then revealed the following week, was new money for the Precision Medicine Initiative for at least next fiscal year specifically. You can see NIH would be getting $200 million more, $70 million of that for the cancer component, $130 million of that for the cohort. FDA and the Office of the National Coordinator for Health Information Technology will also be getting some resources. This, of course, is in the president's budget, but of course, Congress has to decide what they want to pass. And so now is the political season where all this is being looked at, hearings are being held, et cetera, and we'll see, hopefully, Congress will come through with money to allow the initiative to be launched. But again, this is just the president's numbers, and we hope that they will eventually be approved. So what's going on? What is the plan to launch this initiative? And let me tell you, there is no plan. We just have planning. We have a basic idea, but believe me, we are a long way from knowing very precisely what exactly this is going to look like. Let me tell you everything I know up until now. All of the cancer work is under the auspices of the National Cancer Institute, and they have an advisory process that's already very invested in this. And in fact, what this $70 million is going to be applied for are things they were already doing, just going to now do them faster and be able to accelerate the pace at which these programs can be launched. I'm not going to say anything more about the cancer component because I'm not involved in it. What I am, in terms of the policy changes, I'm aware of, but again, not directly involved in it. I would just say keep your eye on a number of things, including a lot of activity you've probably heard from recently going on with the FDA, in part being catalyzed by these kinds of inclusion within this initiative. And I would also note, keep an eye on things being written. For example, Eric Lander wrote a piece in the New England Journal not many weeks ago discussing some of the issues regarding regulating genomic testing in the era of precision medicine, again, to try to help catalyze advances here. What I'm most involved in and will be spending quite a bit of my time in the coming years on relate to the building of this national cohort. Recognizing that this is an audacious goal and that this has to be done right, we quickly convened a workshop to sort of look at some of the things that needed to be done. Actually, before I get to the workshop, sorry, I'm going to tell you what are the elements we know already that we want to have, this idea, the early vision for this cohort, have it be something like 1 million or more individuals, but not to do this de novo, but recognize that there's a large number of existing cohorts, many of which have been funded by NIH, but even when you cobble together existing cohorts, inevitably we're going to want to be able to add additional individuals. So there'll also be new volunteers, a lot to be determined what exactly this looks like. All the participants in this national cohort are envisioned to agree to share genomic data, lifestyle information, biological samples, all linked to their electronic health records. The idea is to provide scientists, of course, to have a ready platform for many new studies to propel our understanding of health and disease, and also forge new models for doing science that emphasizes engaged participants and open, responsible data sharing, but also have, of course, strong privacy protections. Now, I should point out the idea of having this research cohort is not a brand new idea. In fact, everything really that is old is new again. About 10 years ago, Francis Collins himself talked about, in a commentary in Nature, a case for building a prospective cohort of something like this to look at genes and environment. At the time, it was deemed to be too expensive. At the time, it didn't get much traction, and to be quite honest with you, it just didn't get very far. So what's different now? Why is it that all of a sudden there's lots of excitement and the president's announcing it basically as an older idea? And I think the reason is there's a big difference between now and 10 years ago. And I would just quickly summarize it by saying that there's sort of 5 areas of advances that really have just changed the picture why a cohort idea is one that seems so desirable right now. I spent the first half of the talk telling you about genomic advance in the last 10 years and Genome Project, understanding the human genome, technologies, and so forth. It's very clear genomics, I actually think, is going to be the easy part of all of this. But the other thing that's changed a lot are electronic health records. 10 years ago, when Francis proposed this, only about 20 to 30% of healthcare providers in the United States used electronic health records. Now it's over 95% in the US. Now, there's lots of issues to be worked out about interoperability and communication and integrating and so forth, but you're starting at a place where a lot more people are getting medical care associated with electronic health records. Compared to 10 years ago, think where technology has come. Think about the rage right now with mHealth devices, your Fitbits and all your other things for monitoring physiology and lifestyle environment. And think about how they're really crude and early days. Can you imagine 5 years from now where those technologies will be? And so the idea is to take advantage of what is a very quickly evolving area. And all these technologies, of course, are communicating with our smart devices, these smartphones. 10 years ago, only 1 million Americans had smartphones. Now over 160 million Americans, by 58% of the adult population, carry smartphones. Could we envision that just almost getting for free lots of data by getting better and better detectors harnessed to volunteers who are carrying around smartphones? That is a vision many people, including people here, believe in, and now seems to be a very unique time to be able to take advantage of that. Another thing that people here I know will resonate with is think about how far data science has come in the last 10 years. Think about what it's going to be 5 years from now and 10 years from now. We are ready for a super big data kind of challenge related to precision medicine, and the timing is really good to really start that process now. And the last thing, and you're going to hear a lot about this as the Precision Medicine Initiative unfolds, is we're at a different time and a different era when it becomes to participants of biomedical research. And notice I use the word partnership because I think there's been a big cultural change that has gone on that we really want to take advantage of. The people that comprise this cohort are not going to be subjects, they're not going to be patients, they're going to be partners in this endeavor. They're going to be engaged from the beginning as part of the research endeavor, and they're going to be treated as partners. And more what we are learning as you see citizen science and consumer empowerment and going on is that people actually want to participate in biomedical research as long as they get to learn about what's happening and as long as they're asked about what's being done and they're told about the results. We want to harness those energies and the social media rage and take advantage of this in a fashion to help build this cohort. Recognizing there are many details, though, to be worked out with regard to participants and recruitment, retainment, consent, and all the data integration, and all the individuals that we're going to need in a multidisciplinary way. We needed a lot of good advice. We quickly convened a first of what will be many workshops. This just happened to be one where we gathered people in the middle of February. About 90 people came in, but 1,700 people joined remotely. This 2-day meeting to start to flesh out the questions that were going to be needed if we're going to think about launching this cohort next year. And we learned a lot, although, like I said, there's a lot more that's going to be learned. So where are we? What are the next steps? And it is very fast moving. Just this week, I believe this week will be announced, if not this week, next, Francis Collins will be announcing the creation of a working group of his highest level advisory group. That working group is going to be chaired by Kathy Hudson and Rick Lifton of Yale University. Kathy Hudson is one of the deputy directors at NIH. And there'll be a group of individuals that will be assigned to very quickly get going to try to flesh out details for launching this cohort. The planning is going to go on the rest of this fiscal year because funding, if we get it from Congress, will come next year, which means we got to get going to figure out how we're going to put out funding announcements. We are expecting this working group to therefore deliver an interim report in September and likely a final report by the end of the year. To accomplish this, there will absolutely be more meetings and workshops, and almost all those will be video cast live so that people can watch in and learn about what's being strategized about and what's being thought about for launching the initiative. It's going to be implemented not by a single institute or even a couple institutes. It will be implemented in a trans-NIH fashion. I, along with Gary Gibbons, who's the director of the National Heart, Lung, and Blood Institute, are co-chairing a group of institute directors that will be overseeing the governance of this and overseeing the launching of it, taking the recommendations of the working group when those come out in September. And the idea is that by this fall, we'll put out the initial funding announcements because if we're going to spend fiscal 16 money, we got to get announcements out this fall. And finally, it's not just us. We're doing this in coordination with many other parts of the government, including the White House, the Veterans Administration, Department of Defense, Office of National Coordinator, and so forth. And finally, let me just say we're going to try to be as transparent as possible as this quickly unfolds. This is a website you may want to become familiar with where we're putting up everything you could possibly imagine about the initiative. For example, here is we get a link to the workshop that I told you about that we had in February. That brings you to another page. You can get to video archives of the entire meeting. You can get to white papers that were put together before the meeting and refined after the meeting. And it also includes a report that'll be going up in the next few days. Meanwhile, the White House has a web page. You may want to look at that as well with videos and lots of other information coming out from the White House's perspective. And very quickly, let me just point out that since the precision medicine announcement, it really has become all the rage. You know, whether it's sort of covers of magazines in our field or even this was a paper that came out or an article that came out in the Washington Post like last week, I think it was, talking about precision medicine. And then I think as Eric pointed out, I know that he was a part of all this, you know, this was not completely independent, that Apple jumped in and talked about the announcement of their new research kit. Associated with some of their iWatch or their smartwatches, and talking lots about how these devices and these applications are going to feed into biomedical research in ways that exactly will fit in very nicely to the kind of vision that we have for the Precision Medicine Initiative. And of course, that got lots of popular press also following this. So my last slide, let me just pause for a minute and just sort of bring it all back to where I started at the very beginning. The very first slide I told you about, I showed you, was what the circumstance was shortly before the Human Genome Project launched. In fact, if you go back almost to the day, it was 25 years. And 25 years ago, it was the spring of 1990, I remember it well, and we were on October 1 going to launch the Human Genome Project. And you know what? We didn't really know how we were going to do the Human Genome Project. We sort of had a vague goal, but had no clue how we were actually going to nitty-gritty actually get it done, map the genome, sequence the genome. It is deja vu all over again, for at least for me and I think others, that now 25 years later, here we are with a big announcement about this thing called the Precision Medicine Initiative. It's allegedly going to start next fiscal year, which is October 1. And we don't really know what we're going to do exactly. We have a vague idea what we're going to do, but we're going to work intensely to try to flesh out early ideas. And then as we march along, we'll rip up those ideas and come up with other ideas. I'm very comfortable with this, having seen the Genome Project play out. And I believe it's going to play out in a similar way, that we can't even imagine what this initiative will really look like over its lifetime. But it's just so exciting to get it started and to have so much momentum behind it, both scientifically and politically. So I will stop there, and I'm happy to now take any questions you have. Thanks so much.
All right, so we're going to want the discussion to be pretty lively here, and so think about your questions, important questions for Dr. Green. But I think I'll kick off the discussion on so we're all pretty excited about this precision medicine announcement, and my thought is more of what's going to be done to harness, the energy on the private sector, where, you mentioned $250 million, but I could name for you 5 efforts that are spending on the order of $1 billion, whether it's Patrick Soon-Shiong and Nant Health and the $1 billion he spent on precision medicine and cancer. There's Google with their Human Baseline Project, and Calico, which is, of course, ramping up to spend $1 billion+ and Apple and Facebook, everybody's sort of getting into this and are going to spend scales of money and not necessarily pay attention to all the academic and NIH-funded things. They're just going to move and try to drive this revolution as well. And how does the NIH precision medicine fit into that, or what's going to be done to sort of make that more synergistic?
Yeah, no, it's a great point. First of all, I should really stress that the amount of funding being put into this in year 1 is a figure just for the first year. I don't think we even have any idea what the needed funds will be going beyond it. We have to be realistic that, as you well know, it is very difficult budgetary times in the U.S. for all things, but in particular for biomedical research. We are all hurting. So, we just have to be realistic and manage expectations of how much money we would get. And even knowing even 5 years from now what this is going to look like, I think is very difficult. I would say, of course, we are very aware not only of some of the private sector activities, but also other countries as well. And I would say, to be honest with you, that actually I think it's one of the things that very much influenced the president to try to launch this was a significant amount of private sector individuals coming up to him and saying, if you kick this off and galvanize it, you will get a lot of people coming in with their own resources, with their own data, recognizing that there is a lot of stuff that if we all would share that data together, everyone will benefit from this. And we've learned this over and over again in genomics that there really doesn't have to be a competition between the private sector and the public effort. The fact of the matter is everybody prospers when the data is shared. And so I think that, I certainly wouldn't pretend to believe that all these private efforts will come in and merge data. I actually believe some of them will. I also think there'll be a lot of individuals, and we're already seeing this, especially on the data science side, who are very excited about jumping in, and including the Googles and the Microsofts, who say the time is right to do this, and they see all sorts of opportunities that if this is built, that the industry will be able to really build on what's available publicly so that everybody benefits.
Yeah, it does seem like the facilitating of the sharing and governance and the regulatory hurdles could benefit very greatly from the NIH efforts. So I think Johan was, if you have a question sort of going up to the mic so that we can, there's this being streamed live and people want to hear the question. So there are 2 mics on either side if you can.
Thank you, Eric, for this very nice overview. I'm Johan Björkegren. I work in the cardiovascular genomic field, and I assume that with genomic medicine that includes also, molecular biology in terms of proteomes or transcriptome and so forth. And from my limited perspective, it seems like you put up on the sort of these areas of research where you think genomic has been the most fruitful so far, and they're all very disease focused. And to me, it seems like in order to really understand the genomics and to me more the molecular regulatory landscapes, we need to understand, how that works in the healthy situation. So, and we don't really know that, do we? How is the liver machinery really working? How are we to understand complex disease if we don't know? We talk a lot about the networks here in this institute. So and then I was sort of intrigued by your cohort because I think that maybe could be used to those efforts, and maybe we have to take one step back and really have an ambition to find out this regulatory molecular machinery for in health before we can really tackle at least the complex diseases as opposed to the rare ones. So what's your comments on that, and what is your institute?
Well, I don't disagree with anything you said. Probably what you pick up from a talk that I give like this is my worldview of somebody who leads an institute that thinks only about genomics. And so the second it sort of starts to lead into, well, how does the liver work, or how does the heart work, or there are other institutes at NIH that are doing that, and what we're delighted to see is that they are increasingly taking on genomic approaches to do those studies. But of course, I don't need to solve that because others are going to, but doesn't mean I don't think it's important. It just means what I try to do and what our institute tries to do is to facilitate these studies that will then be taken up and looked at in organ or physiological system-specific ways or disease-specific ways. And I think the same thing would be true about the Precision Medicine Initiative as well.
But I think, really to achieve the goal of precision medicine, this is a key step because, we want to translate genomic knowledge into the clinic, but I think this is something we need to. So if your institute is pushing for precision medicine, I think this would be something that would be important too, not just focusing on, quick. I'm with everyone to try to get this to the benefit of the patients, right. But I think we also need to understand that there is a physiology of genomics that I think needs a lot more attention than it does.
No, I think it's a fair point, and I would say that some of the early response that we started to hear about the Precision Medicine Initiative was there really going to be a place for basic scientists to really take advantage of it? And I actually think there's incredible opportunities. I would think if that data sets are available, it would lead to hypotheses being able to be studied by basic scientists independent of clinical care issues.
And hopefully in the 1 million plus cohort, there'll be some individuals. Hopefully.
Actually, I should stress that what came out of the workshop is clearly one of the things that probably will be, not exclusively, but probably heavily emphasized early on, are in particular cohorts where taking people who are getting all of their health care in one system, so that you could have sort of all data, all within one electronic health center. And so that's all comers. So it's going to certainly be healthy individuals.
Perfect.
But to Eric's comment, it's not that easy to sample healthy people. I mean, we do get most disease for obvious reasons.
Yeah, but I think like Eric was saying, if you're sampling over 1 million people from systems that take all comers, there should be a percentage that we can declare as well. Judy.
Judy Cho, genetics and GI. So you made mention of dealing with the roadblocks. I normally don't like to think about kind of dealing with the roadblocks, but this hits me as being essential in terms of the merging to the electronic health record, and kind of in terms of sharing data, perhaps the most important idea of sharing data is how you share, how patient private medical information gets shared. So how are you thinking about dealing with the EHR issues and sharing that information? And because there are some pretty significant privacy, and it's going to change the way how medicine is practiced.
Yeah, so I don't have answers, in part because there aren't answers yet, and it's not an area of expertise I have because I know there's a lot of sophisticated aspects of both the how electronic health records operate and also some of the issues around privacy and exchanging data. What I will tell you is that is an incredibly high priority. It's why incredible high priority with, I guarantee you, among the working groups being set up, and some of the earliest workshops are going to be precisely around that. And it is clearly something that this entire thing will fail if we don't get that right. And we also know that there's a lot of examples with especially interoperability where it hasn't gone right. And But that said, this has attracted a lot of very talented people who want to get involved that we believe will be very helpful to the process.
Again, an example where a government-led initiative is actually going to be crucial in this regard.
Okay, Rad.
Thank you, Dr. Green, for the overview. And following on Dr. Cho's question, I'm Rad Savik, pharmacologist here in Genetics and Genomic Sciences, but I'm curious about partnership with patients. Can you speak to the effectiveness of programs that you have to inform the people and have them, A, comfortable with getting the data and the results, then comfortable with having their own cells maybe existing out there for perpetuity and being used for research, and also keeping in mind the more litigious people as well as their concerns about issues with healthcare, et cetera?
Yeah, well, what I would tell you, we've been involved in some studies that clearly have queried individuals. What is it that people want and don't want? And how much do they want to participate? And I will also say this is probably also influenced by hearing from self-aggregated rare disease communities that have formed and aggregated on their own. And people repeatedly, the data consistently shows that most Americans want to participate in biomedical research. But they want to do it on terms where they feel that they are part of the process as opposed to just simply being sampled and then never hear from again. Now, that doesn't mean everybody has to do it, and so there has to clearly be the ability not to participate. But it is very clear that there's a thirst out there, and there's been a number of movements, citizen science-like movements that have clearly shown there's a lot of energy out there from disease communities of various flavors and even just from the general public. And we believe if we approach this right, and I will also say, probably thinking sort of about, what's the movement, social movements going on, and where are they likely going to be 5 years from now? We think there's a lot of momentum that a lot of people will. And I would also point out, this is not just based on us believing this. There are examples, and I would point to some healthcare networks, for example, Geisinger, Kaiser, some other major networks where they have launched things very similar to this, and their uptake is actually quite high. But you absolutely have to build in all the right privacy protections. You have to do this in terms that patients can opt out at any point. But I think we can do this right, and I think people will participate.
And people do consent to Gmail, being read by Google, and you divulge lots of pretty highly personable things in your emails, and that can be mined and used for various purposes. So I think what we can return back is the service that's going to be offered back to people actionable enough, informative enough to where they tolerate some loss of that.
One of the things that got discussed at the workshop, for example, is, people actually are the studies show people are very frustrated by healthcare systems that allegedly use electronic health records, but a patient can get no access to their own information. Yeah. Imagine the process of fixing this so that we can get all this electronic health data into some central knowledge base. We also make this so that patients can access their electronic health data that they own. It's their data. They can get on their iPhone. That'd be pretty cool. And that would, that's something that we should strive to do. Right? Right.
Yes.
So that's part of the social movement. Right.
So I think we'll do Ron first and then Pamela.
I'm Ron Chen in clinical genome informatics, big data. So I'm very happy to see you have at least 1 million genome cohort. So far I already have 100,000 whole exons, but the phenotype has been very limited, only describing of the disease. So the missing data is really on this very detailed phenotype. So I'd like you to seek your comment on two questions. One is about data sharing. So I have at least 100,000 exons. Many people reach me to access it, but because I get from dbGaP, I'm not allowed to release it. I'm wondering from NHGRI, how you encourage people, like at least unblocking us to release the data. And second is, so in terms of this 1 million genome cohort, how you select these ones, you touch on need to be under the same systems. So what we already have here is we have 3 million people with last 10 years of longitudinal full electronic medical record. So including 1,700 gene knockout people, each of them has 6,000 lab test results. So I'm active mining on that one. I'm wondering whether it's selecting some large hospital with a very comprehensive longitudinal phenotype data to focus on this 1 million, or is it just selecting from the whole world? It's very diversified. How do you combine all these phenotype?
Yeah.
With wrong PIing the grant.
Oh, I see. That was a big part of it. The answer to your second question is all to be determined. I mean, so that's exactly what this working group is going to do. There's going to be some sort of initial idea of blending by some criteria, bringing people in. But what that looks like is, I wouldn't even venture to guess, because that needs to be intensely looked at to decide exactly, which individuals from which healthcare networks, and inevitably that won't be enough. How will we decide who else is in, and so forth. In terms of your first question, the model that is envisioned initially, although again all to be determined, is sort of a DB gap-like model. Have the data come into some place that should be shareable if you are sort of you have to apply to be able to get access, to be a legitimate researcher, et cetera, because it'll involve clinical data, it will involve a lot of personal data. That's envisioned to be the model, but to be determined exactly how we might refine it going forward.
And I think part of the question, though, was do you envision, if that is a system that will be adopted, a more efficient way to, for those who are approved to look at such data to better open sharing so that we're not all filling out these applications. Yeah.
Needs to be looked at. It's a fair point. We know that needs to be looked at.
So I have actually sort of a simple question that follows up Vlad's comment. Because it always seems to me the devil is in the details in terms of, we all agree it's nice to give back information, it's nice to share information, it's nice to enable people. But I found recently on a variety of boards, when I suggest let's give the patient back their actual data, people are horrified. The researchers are just completely opposed to that. They may be willing to give back, actionable variants or something like that, but simply giving your test subject a USB with their own sequence, just like you might give them a copy of their X-ray as part of a research protocol where individuals don't normally have any rights to actually access their data. I'm curious what your personal thoughts. I know it's not been worked out yet in the sort of the larger project, but I think that's where the public is. That's presumably what they want. That's what I would want. That's what I advocate for, and I don't see that being sort of mainstream among the research community.
So I guess my personal view is I just think it's their data. And so I think maybe that's one of the key rationales for doing this is that it would be codified, that if you participate, you have access to your data, and there won't be researchers standing in the way saying, no, that's my data. I'm not going to let the participant get to that because I don't want to.
So that's the plan for this project.
Again,
or that's what you'd like to see.
I'm giving you my opinion. I can tell you the spirit and the philosophy
right.
that's coming out of this is that to do, and the surveys and the interactions with participants seem to bear this out, that you will get buy-in and you will get people to participate as long as you treat them as participants and they have access to all of their data and all the resulting analyses that are done with their data.
Yeah, I think that it'll be a tough go with the academic community getting that. It would be great if you have, but that's
and what I would say is that there were lots of reasons why we believed that the Human Genome Project would not be successful, because we needed cultural change, and maybe what we're looking at is the cultural change that's needed in the scientific community in the next 5 years.
So I think, was it going to be Andrew or Boris?
Whoever asked the easier questions.
So this will be fast. I don't know if it's easy. So you mentioned obviously that it's very easy to generate genome data, and with the advent of various monitoring devices and the pace of innovation there, it'll be pretty easy to get data on how people are functioning over time. Of course, we know that a lot of human phenotype is a consequence of both genes and environment, and what seems to be missing in many cases is actually good environmental data, good environmental sensors. And I'm just wondering if you've had any discussions with other agencies in the federal government about how to actually access and get that type of information accessible for research as well. Yeah, no, it's a great question, and some of this is fingers crossed. There seems to be technology surges everywhere, and wouldn't it be great if there was similarly, imagine if we could see over the next 5 years technological surges for measuring environmental exposures that we've seen in DNA sequence for the past 5 years. That would be awesome. We do have some areas of NIH that focus on that, and National Institute of Environmental Health Sciences. There are other parts of the federal government that are interested in this, and some of them are being enlisted. And I think the private sector is very motivated to do this as well. So I don't have any particular insights about this except just sort of fingers crossed and some early, some people telling us there's some promising developments. Well, we're very interested in that here. We've got an environmental Edison Institute, and that's a matter of great interest. So,
Right.
Bruce tells me I asked his question. So.
Okay.
Over to Boris.
All right, Boris.
There are a couple of times we heard the word phenotype. And my question, all this project will be successful if we will have the formalized phenotypes, formalized diseases. So do you have any idea how it will be done for 1 million people? Because we need records for this, and how it will be measured.
Well, I mean, so I think the things I would say would be having access to electronic health records will provide some data, not perfect, but some data, and we're getting better and better at mining that data. Phenotypic data can come from some of these health devices, especially if they get to be research grade as opposed to recreational grade, which is sort of where a lot of them are at right now. And then we have the ability to imagine studies that would be done. Once you have sort of a basic cohort, you may say, hey, these 5,000 people would be really interesting to study in greater detail based on some preliminary genomic data and other and electronic medical data. And then you bring them in and bring them to study them more intensely for some phenotypic characteristic.
May I have a question? One more.
Okay, Boris gets 2 questions. Everybody else gets 1.
Yeah,
So it better be good, Boris.
Okay, it's great to have this precision medicine approach, but what about precision psychology and studying the human behavior? So really to discover the phenotype and being able to select mathematicians or musicians early and give them the chance to get developed.
And the garbage collectors too, Boris?
Let me just say I am quite sure, as the director of the institute that has led the ethical, legal, and social implications efforts in genomics, there will similarly be major bioethics questions to be studied as part of this, and probably it's a good idea. More seriously, to answer your question, I mean, obviously all these things would be very interesting as well. I'm sure there's a lot of challenges with phenotyping behavioral characteristics as well. But you know, certainly this could be considered for the future, but I'm sure those are going to be quite challenging.
All right, I think Erwin was.
Yeah, Eric, thank you for this overview, and had the privilege of being at the workshop, and it was incredibly exciting.
There you go.
And I think, you know, you and the Institute, NHGRI, need to be complimented. Excuse me, I'm German, so I get my I's and E's not always straight. But for actually taking on the implementation issue. And I know this has not always been an easy discussion at NHGRI and Council, et cetera, and there are different opinions in the genome community. But implementation gives us the opportunity to learn so much. And as you pointed out, we are you know, privileged to be funded with the Ignite Network grant and the eMERGE grant. And I can tell you with regard to patients and the people in the community wanting their data and actually being excited about it, that after having 1,000 people enrolled now in our implementation programs, patients and people from the community love it. They really are feeling empowered. They say it makes a big difference if I go to my doctor and I can tell them this is what matters to me because this is in my genome. And so going forward, I think one of the important aspects is to be inclusive and to have a broad representation of all populations in this country in the exciting Precision Medicine Initiative. And when you mention there are certain institutions like Geisinger and others, compared to our institution here in New York, it's almost a mirror image in terms of uniformity versus heterogeneity. So what are your thoughts and the workshop thoughts on making sure that the people from El Barrio here, our Hispanic patients, and from Harlem, our African-American patients, are represented at the table at this initiative?
Well, what I basically would almost echo what you said, is that it was a recognition we have to be incredibly conscientious of thinking about what this looks like. You know, I bet at the end of the day, 1 million is going to be too small. I bet it'll be bigger, and I bet we're going to have to figure out, you know, sort of what the right way to blend different, different cohorts or recruit to fill in to make sure we have adequate diversity and adequate representation. Exactly what those numbers look like and exactly what that, that has to be worked out. And it probably won't be perfectly worked out in the first year, maybe not even the first 2 or 3 years. But we should be conscious of that absolutely at every step along the way if we're going to do this right. And the U.S. is a big, complicated place with lots of different populations, and we need to make sure all of them somehow work their way into this cohort.
Hi, William Mindich, Children's Health and Development. So I'm kind of interested to hear how this coincides with other really large genomic announcements like the 1 million veterans, and how much collaboration there's going to be between that and also the open source community that, for example, with 1 million veterans, is doing a lot of work with their EHR.
Yeah, so I can tell you that from the very beginning of the initial discussions in the President's office, the Million Veterans Program has been discussed and is actively being consulted and thought about exactly how it will blend into this. That's why I say NIH is not doing this alone. And there are some unique issues about how that was set up and exactly under what terms the people agreed to participate. This is not an obvious exactly how to proceed. But there's no question there's not a meeting that goes, major meeting, including this workshop that goes by, that people in the Veterans Administration are not involved at the highest levels to see how this might synergize. Similarly, there's parts of the military, and the Department of Defense is also involved in this because the military has significant interest in doing these kinds of cohort studies and exactly what that looks like and how they participate, but I'm sure there'll be components of that as well. And also, I should stress, internationally. There's the UK Biobank, there's Genome Tank, there's a lot of other issues, programs in Estonia. So we all, in fact, some of those are individuals who we're consulting with, especially the UK Biobank. They have a lot of experience. The US is a little late to the table here. There's other countries are pushed, but we'll catch up. But we can use their expertise to help us in some of the early stages.
Okay. Jonathan, were you poised to?
Jonathan Carr, Genetics and Genomic Sciences. Very excited about the Precision Medicine Initiative. So I'm curious from your perspective or from the perspective of the planning committee, how far along do you think precision medicine is compared to, say, the Human Genome Project? Is precision medicine where we were in the Genome Project in 1990? Are we 10 years away from having a draft of precision medicine? Yeah. How far away do you think we are?
Well, so first of all, I acknowledge that the analogy between the Genome Project and the Precision Medicine Initiative falls a little bit down when you recognize that the Genome Project had a very crisp endpoint for the most part. Sequence the human genome and you're done. And so whereas exactly what the endpoint is in the precision medicine is obviously it's not as crisp. To answer your question more directly, I think, first of all, I think in some cases, for some early applications, we're there with precision medicine. There are some very clear indications in cancer and pharmacogenomics and some of the things I listed as hot areas. But I think it's like the very tip of the iceberg. Where will we be 5 years from now and 10 years from now? I'm not sure. I guess there's probably people here as qualified as me in their area of physiology or disease probably can even give a more informed explanation. I personally believe that we will see systematic advances every year over the next 15, 20 years, maybe beyond. And so I don't even have any idea how long this initiative will go on. I think to get the full impact of a longitudinal cohort, I think we should be prepared to be doing this for a couple decades at least. So I think it does differ from the Genome Project in that it may go much longer and will morph probably multiple times along the way.
Ray?
Hi, my name is Ray Chang. I'm with Genetics and Genomic Science. So very exciting presentation. And my question basically is that to me, it seems like the driving force under the precision medicine is basically one part is methodology development, the computational methodology development. We all talk about the data generation. We have generated a lot of data, and then now comes to the part that we need a method to analyze. So from your talk, it's not very clear to me like how much emphasis in your budget that will be put to the new methodology development? Because basically the new methodology development will cost a lot of time, will be a high risk but high return. But basically, I didn't see this here. And another question is that from the NIH announcement, like the RFAs or the PAs, the RFAs, basically there are not very much explicitly RFA is stressing that calling for new method development on the computational new computational method development from your institute. I see a lot of from other institutes, but not from your institute. And probably also that the computational approach has different levels, like from the very broad bioinformatics to the very deep down network modeling stuff, which is more difficult and costs more time. So what is your comment on this?
Well, when it comes to Precision Medicine Initiative, there's nothing determined in terms of budget below sort of an indication of what we think we'll put into the cohort in year one. What that looks like in terms of division between developing technologies, doing data analysis, recruiting people is all to be determined. Again, this is all we're at the earliest stages of planning. So that's why I haven't said anything about, because I have no idea what it's going to look like. In terms of the comment coming back to either what our institute's supporting or other parts of NIH, what I would say is there's actually a lot of that kind of development going on in the Big Data to Knowledge initiative, BD2K, where we are, that's sort of pooled NIH-wide money we contributed to, every other institute contributed to. So we're seeing that's our attempt at a corporate level to sort of jumpstart some of that. That said, I agree with you. If I wish we had more money, I wish we should be doing far more in data science than we are. Money's really tight, and so budgets are tight. We can't do everything. But I completely share any frustration you may have that there's not more NIH money being put into data science. I've been one of the strongest advocates for data science at NIH, which is what led to the BD2K initiative.
Right. I simply just want to say that the method is also important, that I have no frustration with NIH.
Oh, I have frustration. I have frustration. I wish we could fund more, because I fundamentally agree with you. I mean, I think the data science part of precision medicine is going to be the pivotal part, and and if we don't fund that area better, we won't get the young trainees here dedicating their careers to it, which is what we need 5 years from now.
Okay, so maybe just 2 more questions. Yeah, start with Zainab.
Hi, Zainab Gümüşin, Genetics and Genomics. We met before.
Hi.
So, my question is, in clinical genomics, especially in cancer, now when we apply these precision medicine approaches and identify, for example, a actionable mutation or a hotspot in a patient in a clinical setting, for example, with the impact panel or the Foundation 1 panel, et cetera, there's still some hesitation in the perspective, point of view of the oncologist, and rightly, because it's a risk to do an alternative treatment for the patient. So what are some of the studies that are being undertaken right now in order to help with the speed up of this process? For example, if it's a rare cancer where there's no precedent, there's a lot of hesitation because it's undue risk. Maybe that hotspot is actually a protective hotspot, but is involved in cancer, for example. So what are some of the initiatives that can help make this process move faster? I mean, the kind of findings that we have in the clinic, in the clinical genomics and sequencing, to move to actually to the patient.
Right,
So.
and you mean?
And then just to complement that too, is who pays for this? It's worth noting that foundation doesn't make money. We're losing lots of money every year, so they can't get these tests reimbursed, and so it's sort of a good, both the actionability of physicians using it, but then also being able to bill and recoup money to be able to run that kind of test in a routine way.
Well, the whole reimbursement arena is a complicated one that has to also be dealt with. I mean, we interact a lot trying to help educate, people that make decisions about reimbursement. But they want more evidence base, which relates to the first part of our question. We need more studies. So our clinical sequence and exploratory research program has within it, I think, at least one, maybe even a couple cancer component projects. We fund that jointly with the National Cancer Institute. I will tell you, the National Cancer Institute is ready to really push the accelerator and put even more clinical studies in play And in fact, some of this precision medicine money, if it comes, will go to accelerate some of that. We simply need bigger studies and generate bigger, more convincing data so that practice guidelines get changed by professional societies.
Is there a repository right now where we can go and look, for example, for a specific cancer in different institutions if people are taking the alternative risky approach as a last resort? Is there a way that we can put this data together so we can know what other studies or what patients have been treated with?
So I don't have an answer to that. If it's around cancer, it would probably the locus of involvement would be the Cancer Institute, not our institute. Maybe somebody here knows what's going on if there's any attempts to do that. But that would be the Cancer Institute.
Thank you.
All right, last question. So it better be really good.
Hello, Roman Kosoy, Genomics and Genetics. Thank you very much for coming.
Sure.
It's very nice to hear you talk. And the question I have is about the public education. It's like with this huge project when we're trying to involve a very large number of people and we're trying to raise enthusiasm for the precision medicine. We need to have a system and some kind of infrastructure which would allow people to interpret their own results. Because right now there is no such way. I have many friends and family coming to me and asking me, can I do this? What does it mean? And at this moment I have to say, I have no idea. Because, I mean, yeah, you can read on Wikipedia, you can read here, you can read there, but there is no centralized system which would actually give you guidelines or any way to connect with publications in such a form that people can understand. And also this kind of public outreach system would also really help to involve researchers connecting with the patients. And would actually greatly increase the political clout of this kind of system and might help with getting additional support for many, many years to come.
No, I agree with you fundamentally. I mean, we have a number of educational programs that we've been involved in, and some of the private sector groups have also included educational programs they've put together. I think there's been a lot of good engagement and collaboration across multiple funding agencies, multiple institutions around education, and I'm sure this will be discussed as this initiative gets off the ground, because I do believe we can't just imagine putting a cohort together without also thinking about how we're going to educate people who are participating in this.
It would be nice to have this kind of infrastructure being set up as early as possible, because it will be adjusted constantly, because seeing how people respond to it, where, which aspects of it they actually want to follow, which not. It's just right now there is a big empty hole where it exists. And 23andMe had a very big following because of this, but it has its own challenges. It would be nice to have something publicly driven.
No, I agree with you.
Thank you.
All right, well, let's thank Eric Green again for a very successful launch of the seminar series. And thank you all. We're very excited about precision medicine and hope to be interacting with Eric in the future.